Welcome to our lesson on DNA replication, which is the copying of DNA. Before we jump in, let's look at DNA structure again and review it briefly. This is what DNA looks like. DNA is a nucleic acid. It's made up of repeating units or monomers known as nucleotides. So this is one nucleotide, this is another, and another. We also know DNA is double-stranded, so there's two rows of nucleotides that make up DNA. Now, one nucleotide is made up of three parts. This is the phosphate group. This is a 5-carbon sugar called deoxyribose, which is what DNA gets its name from. DNA is deoxyribonucleic acid. And the third part is this nitrogenous base, which comes in 4 different flavors: adenine, thymine, guanine, and cytosine. And we use the first letter, A, T, G, and C very often to represent them. So the backbone of the DNA, the outside of the ladder, is made up of sugar and phosphate, and the inside is made up of these nitrogenous bases that are responsible for the DNA sequence that makes each individual unique and different. The nitrogenous bases form different base pairs, and C and G's always go together, and A and T's always go together. All right, the other thing we learned about these bases is that some of them have double rings, like this one, and some of them have single rings. The single ring ones are called pyrimidines, and they include thymine and cytosine, and the double ring ones are adenine and guanine. Now, the DNA becomes twisted up into what's known as a double helix or twisted ladder. So that's a brief review of DNA structure. So why would we want to copy the DNA? If you remember in the previous chapter, we were learning about mitosis. Specifically, we were learning about interphase. And we know that in interphase, the cell grows, it does its metabolic function, and it also copies DNA in a process known as DNA synthesis. DNA synthesis is what we're looking at here. This is when the DNA gets copied and your cell is getting ready to divide, and each new cell is going to need its own copy. So this is where DNA replication occurs. S phase of interphase. Now, chromosomes replicate in their entirety, resulting in identical chromosomes, right? And in eukaryotes, we know that these copies are called sister chromatids, and they're held together with the centromere, and they're attached until they're pulled apart in anaphase. All right, but the copying of DNA replication is today's topic, and that's something that happens in the S phase of interphase. Okay, this is how DNA replication occurs. The first thing that has to happen is you have to unwind the DNA. It's very twisted, and we're going to unwind the DNA, and there's an enzyme that does that. It's called helicase. The helicase also not only unwinds the DNA, but unzips it. So it actually, like a zipper, it will unzip the 2 strands of the DNA from each other. Now, there's also other proteins called DNA binding proteins, or SSBs, that keep the strands separated and untwisted because the DNA really wants to get back together. The 2 halves want to get back together with each other, so these SSBs keep the 2 strands separate from each other. The next thing that happens is something known as a primer comes into play. And the primer is going to base pair with the exposed single DNA strands. And this primer is really necessary. It's made up of RNA, which we'll look at later. Primer is a short single strand of nucleotides, and it's going to allow the DNA polymerase, the main enzyme for DNA replication, to attach. So you need this primer to get the whole replication process started. So this primer is going to come in over here. The next thing that's going to happen is DNA polymerase, which is indicated with this green box. This is an enzyme. Remember, anything that ends with ASE is an enzyme. And DNA polymerase is actually going to build the new strands of DNA by bringing in more nucleotides. So starting with the primer, DNA polymerase will assemble new strands of DNA from nucleotides using the parent strands as templates. So we're going to use these strands as templates. If there's an A there, DNA polymerase is going to bring in a T. If there's a C here, DNA polymerase is going to bring in a G. And it's going to start building the second strand of DNA over here. Now, DNA polymerase, of course, follows base pairing rules. T is added when it reaches an A. A is added when it reaches a T. G is added when it reaches a C. C is added when it reaches a G. So you're going to start building DNA strands here, and you're going to start building new strands here. So if you can see what's happening here is these are the old. The blue is the parent strand, and the red is the new strand that's being built. And the new strands are being built by DNA polymerase that's actually bringing in new nucleotides here to assemble the new DNA. Okay, so what happens here is an enzyme called DNA ligase is going to seal any gaps that remain in the DNA. So you can see here there are these gaps, and this DNA ligase enzyme is going to come in here and make sure that we have smooth and continuous strands of DNA. Now, each parental strand serves as a template for the new strand. So the old parent strand is like a template or like a model, and each new DNA here comes from one template strand, so one from the parent, and one new strand, which is the red one here. We call DNA replication a semi-conservative process because you're kind of conserving something from the parent DNA, but you also have something new, so it's semi-conservative. Okay. So if you remember, we discussed that DNA is anti-parallel. So it has a 5' end, it has a 3' end, and the other strand of DNA is upside down, right? So it's going from 5' to 3' like this, or you could just say it's coming from 3' to 5'. So this presents some challenges during DNA replication. All right, so DNA polymerase, which is that cute little green enzyme that builds the new DNA, it only works from 5' to 3'. It attaches nucleotides to the 3' end, right? So it's only working like this. Here's the 3' end of the parent strand, and it works in a 5' to 3' direction as it's building the new DNA. The problem with that is only one part of the DNA can be assembled continuously. This is called the leading strand here. So here's what happens. The DNA is unwound. This is the replication fork. This is the origin, which is where you're starting replication from. And we're starting here at the 3' end. DNA polymerase is going to attach and build this nice, long, continuous strand. The problem is on the other side. This is called the lagging strand. And what happens here is we still can only go from 5' to 3' end, but that would be in this direction. So the problem though here is that the DNA is a little bit unzipped. So you're gonna build a part right here of the DNA, and then you're gonna unzip the DNA more, and now you have to build a new segment from 5' to 3', and then the DNA is going to unzip some more, and then you're going to build this part, 5' to 3', and it's going to unzip some more, and you're going to build again in this direction, 5' to 3'. So what ends up happening is you have these little pieces of DNA called Okazaki fragments. You can't go in a continuous direction. As you unzip, you can do a new piece. And these, on the lagging strand, you create these fragments, and they're called Okazaki fragments after the scientists who discovered them. And these pieces are not continuous, and this is where you're going to have that DNA ligase come in and join the pieces to each other to form a continuous strand like you have on the leading side. Okay, and look down here at how this is happening with the base pairs because it didn't have such a clear picture last time. When you're building DNA, you have the template strand from the parent, which has all of its T's, G's, A's, and C's, and this is the new strand on the right that's being built, and DNA polymerase is bringing in a nucleotide with a C because there's a G here, and it's bringing in a nucleotide with an A because there's a T over here. So nucleotides are being brought in by DNA polymerase to form this new strand over here. And again, DNA is semi-conservative because you have this strand from the original parent DNA, and you have this strand that's being assembled by DNA polymerase, which is a new strand. Okay, here's another picture of how DNA replication is happening. Here is your leading strand on this side, and you can see it's being built over here, okay? And you, on the other side, you have the little pieces in the lagging strand, the Okazaki fragments, which need to be joined together. Okay, so one more thing to talk about. As the two DNA strands open at the origin, these replication bubbles form. So not the entire DNA becomes unzipped at once. It's just pieces of the DNA that become unzipped because remember, DNA is extremely long in comparison to how wide it is. So here you're going to unzip a little bit of the DNA, and you have these formation of these things known as replication bubbles. And this is where replication is going to occur. And that bubble moves until you form copies of the whole entire DNA, right? So the replication forks extend out, and you're going to copy all of the DNA eventually as these bubbles move throughout the DNA strand. So in eukaryotes, we have more than one replication bubble. We have many. And in prokaryotes, since they're so simple, they just have one replication bubble that moves throughout the whole single chromosome in a prokaryote and allows them to make 2 copies of their single chromosome. Okay, so there's mistakes in DNA copying. As you can imagine, DNA is very long, so it's very easy to make some base pairing errors. So what happens here is DNA polymerase makes mistakes. It makes about 1 in 10,000 base pairing errors, which is a lot, which is very scary. However, DNA polymerase also has a proofreading function, and it can check its work and go over it and fix any of the mistakes. So for example, here, A and T seems fine, that goes together. G and C is fine, that goes together. A and T again, that's fine. And then T and, oh no, there's a C here. This should be an A. That's a mistake. That's a mismatch, right? That's an error that needs to be fixed because errors can lead to mutations, and mutations could lead to things like cancer. So we have to fix that. So DNA polymerase would go and proofread and replace mistaken base pairs like this. And after proofreading, the error rate becomes 1 in 1 billion, which is much, much better. Okay, finally, chemicals, UV rays, carcinogens, things in your environment that aren't good for you can damage DNA, right? They actually cause these mutations to occur in your DNA. And your cells are constantly having to fix that DNA. So if you go out into the sun without sunscreen and you're exposing yourself to sunlight and you're becoming burned, you're damaging that DNA, and your body's on like overdrive trying to fix all those mistakes in your DNA and keep you healthy and safe. Excision repair occurs when any of over 50 repair enzymes remove damaged parts of the DNA, and DNA polymerase and DNA ligase replace and bond the new nucleotides together. So what happens here is you have a UV radiation produces a thymine dimer, and what happens is we notice this mistake over here. So enzymes are going to cut the damaged region of this bubble, and DNA polymerase replaces the cutout DNA and puts, and the DNA ligase will seal it and fix it. So here we have a DNA that has been repaired by these enzymes. Unfortunately, we have this repair mechanism built into our cells in the form of these enzymes. So I hope that was helpful. It was a good introduction to DNA replication. Thank you so much for watching and listening.